Endocrine system. Endocrine system session learning objectives. Describe the general features of endocrine system, including how hormones are transported between an endocrine gland and its target cell. Name the 3 structural categories of circulating hormones and give examples within each category. Compare the transport of lipid-soluble hormones with that of water-soluble hormones. Describe the specific structures associated with posterior pituitary and anterior pituitary. List the hormones released from the hypothalamus that control the anterior pituitary. Explain how the hypothalamus controls the release of hormones from the anterior pituitary. List the 2 specific types of endocrine cells within the thyroid and specific hormone produced by each. Describe the structure and location of the adrenal glands. Describe the homeostatic system involved with cortisol. Compare and contrast the primary types of pancreatic islet cells and hormones they produce. Describe the action of insulin in lowering blood glucose concentration. Describe and explain the action of glucagon in raising blood glucose concentration. And describe the general structure, location, and function of the parathyroid glands. Alright, let's get started. Endocrine overview. The endocrine system is composed of glands located throughout the body that synthesize and secrete hormones. It includes a cell, termed target cells. It has specific receptors and specific hormones. Endocrine glands lack ducts, unlike exocrine glands. The hormones are compounds that exit into the bloodstream through capillaries, and they enter the interstitial fluid, where they can potentially affect all body cells. However, the hormone only binds to its target cell, which have receptors for a particular hormone. The receptors may be found on the cell membrane or inside the cell. When the receptor is bound, it will initiate or inhibit selected metabolic activities within that target cell. These will then alter metabolic activities that have long-lasting effects and may continue after the removal of that hormone. It's important that physicians and doctors can measure the blood hormone levels to determine certain conditions of the body. The endocrine system regulates development, growth, metabolism, maintains homeostasis of the blood composition and volume by regulating concentration of specific substances in our blood. Here is a list of all the key glands, as well as some of the main hormones to be aware of. In the pineal gland, we have melatonin. In the thyroid gland and parathyroid glands, we have thyroxine and parathyroid hormone and calcitonin. In the stomach, we have gastrin. In the pancreas, we have insulin and glucagon. The kidneys produce calcitriol and angiotensin II. The intestines secrete secretin. Testes secrete testosterone and inhibin. For female ovaries, they will produce estrogen and inhibin. The adrenal glands will secrete steroids and catecholamines. The liver creates EPO and IGF. And the heart produces atrial natriuretic factor. The adipose tissue creates leptin. And the hypothalamus and pituitary gland can release some of the key releasing and inhibitory hormones, which we'll talk about. Patterns of organization in the endocrine gland. Endocrine cells can either be organized as a single organ with only endocrine functions or small clusters within other organs. Endocrine glands are typically composed of a connective tissue framework, which houses and supports epithelial tissue that produces and releases hormones from their secretory cells. The 5 major endocrine organs that we just talked about include the pituitary gland, the pineal gland, thyroid gland, parathyroid gland, and adrenal gland. Endocrine cells that are housed within other organs are like the hypothalamus, the skin, the thymus, the heart, the liver, the stomach, the pancreas, the small intestine, the kidneys, adipose, and also the gonads. The regulated secretion of a hormone from an endocrine gland is controlled through reflexes that require different kinds of stimulation. Some could be hormonal, humoral, and even nervous-based stimulation. So you can see that this is a cell that releases a hormone that transports in the blood and it binds to its target receptor, which will then elicit a response. Categories of hormones. All circulating hormones are synthesized within the endocrine cell from either cholesterol or amino acids. Steroid hormones are generally lipid-soluble molecules that are synthesized from cholesterol, and they include the steroids that are produced in the gonads and the adrenal cortex. The other peptide base are going to be biogenic amines, monoamines, or modified amino acids that are water-soluble. These ones are the ones found in the adrenal medulla, thyroid hormone, and melatonin. However, it should be noted that the thyroid hormone, thyroxine and triiodothyronine, is a peptide but is considered lipophilic. All the other ones will follow the normal rule, meaning if it's steroid, it will be lipophilic. If it's peptide, it will be hydrophilic. This is the only one that does not follow that order. The target receptor is also different for different kinds of hormones. Some could be found in the cytosol, some could be found in the nucleus, some could be found in the cell membrane. Here are some examples. Estrogen binds to the cytosol. Thyroxine triiodothyronine targets the nucleus. Adrenocorticotropic hormone, ACTH, targets the cell membrane. Growth hormone targets the cell membrane. Adenosine triphosphate targets the cell membrane. Prostaglandin E2, PGE2, targets the cell membrane. Nitric oxide, NO, targets the cytosol. Acetylcholine targets the cell membrane. Some of these chemicals are considered local hormones, which are a large group of signaling molecules that do not circulate within the blood. And instead, they bind to the same cell that produces them. They're either close by, called endocrine stimulation, or paracrine stimulation. For example, the prostaglandin activities include stimulating the hypothalamus to increase body temperature, cause fevers, inhibit stomach secretions. It can also act on mast cells that release inflammatory chemicals. The general property of hormones include that they're regulated via physiological processes. They have a variety of chemical structures as mentioned. They're released in small amounts and highly regulated. They move through either diffusion or through the membrane. They can bind to a target receptor located inside the cell or on the outside of the cell. And they're released based on disturbances and changes to your homeostasis. The number and types of receptors in a target cell alter the effect of hormones. Number of receptors. The number of available receptors in a cell fluctuates. Increasing the number of receptors is called upregulation. And this increases sensitivity to a hormone. Decreasing the number of receptors is called downregulation, and that decreases sensitivity to a hormone. Therefore, changes in receptors also occur as a consequence of developmental maturity. For example, the cell's state of activity, the stages of the cell's life cycle. Over here's an example. Let's start off with a hormone A coming in, which will then increase the sensitivity of those receptors, creating more of these receptors on the membrane. As more chemicals bind to it, it's going to cause it to downregulate, therefore reducing the number of receptors only to 3, and now slowing it down. It will then break down those receptors so that it keeps the cell regulated. Permissiveness is a process by which hormones are regulating the receptors based on levels of other hormones. And in some cases, a hormone can even regulate its own receptors. Hormone release is an amplitude modulated. Neurotransmitters release is all or nothing, meaning if you have an action potential, it will cause a release of the vesicles. Hormones, however, never approach 0, but they can fluctuate between high and low ranges. So you'll see over here that a normal physiological range can go up and down like a wave. A range for another hormone could be even more narrow, go up and down, up and down, up and down. But you can see that the range is a lot different. For the hormone number A, it can range between 10 microgram or 10 milligram or 10 nanogram, all the way to 100, whereas the hormone B can range between 40 to 60. So that precise window by which the amount is available is very critical. Peptide and protein-based hormones are synthesized via an active form. This means that the hormone starts off as what we call a pre-prohormone form, which includes genes for different chemicals, the RNA coding for a signal peptide. You may have your target hormone, the antidiuretic hormone, another hormone, neurophysin, and a copeptin. And when this hormone gets processed, it becomes a prohormone. The prohormone will be modified through proteolytic cleavage and becomes a final hormone in which that one hormone gets produced. Most of these hormones are encoded by genes in which they're close to other genes that code for other hormones. So a signal peptide is very critical for directing the hormone to the correct intracellular organelle for processing and creation. Here are the processes that influence hormone secretion. Number 1, it depends on the physiological environment, number of ions available, and what conditions the cells are in. Number 2, it also receives direct input from the nervous system and also neural-based hormone release signals from the brain. It also responds to other hormones in the nearby proximity, and it also responds to mechanical stress or cellular metabolism. Hormone transport within the blood and are carried to the target cell. Transport in the blood. The transport of lipid-soluble hormones is dependent upon the water-soluble carrier proteins synthesized by our liver. Your liver plays a role by creating these carrier proteins that are very selective. They bind to only one specific lipid-soluble molecule, while others could be non-selective, which can bind to several different types of lipid-soluble molecule. Any hormone that is attached to a carrier is a bound hormone, whereas if it's not bound to a protein, it's called a free hormone. Only a free hormone is generally able to exit the blood and bind to the target organ. So you can see the fat-based soluble hormones have to be bound to a transport protein, less than 1% is free, and the water-based soluble hormones are dissolved inside the blood and they're 100% free. Hormones are excreted in the urine or the digestive tract. The water-soluble free hormones are smaller than kidney filtration barrier, therefore they get excreted out in the urine. Lipid-soluble hormones are excreted in the urine if chemically modified to be more water-soluble, otherwise they're excreted through the GI tract. And the half-life represents the amount of time it takes to reduce it by 50% inside the blood. Hormones exert their physiological effects as a result of the blood concentration and have to be tightly regulated. Hormone release and hormone elimination can affect the amount of hormone concentration. It should be remembered that water-soluble hormones generally have a very short half-life, whereas lipid-soluble hormones have a long half-life. Sulfonation and glucuronidation are two common processes by which the body detoxifies and eliminates waste. This includes drugs and environmental toxins. Sulfonation involves an addition of a sulfate molecule to a substance, and this process is catalyzed by enzymes called the sulfotransferases. And as a result, it makes something a little bit more polar and more water-soluble than the original substance. And it helps it to be eliminated from the body. So you can see estrone, which is a version of the estradiol or female sex hormone, gets modified to estrone sulfate so that it can be easily modified and is secreted. Glucuronidation involves the addition of a glucuronic acid to a substance. This process then is catalyzed by another enzyme, which is called a glucuronyl transferase, which transfers a glucuronic acid from some donor molecule to the substance being modified. And this, again, also makes it a little bit more polar as well as more water-soluble and helps it with elimination from the body. Hormone receptors. G-protein coupled receptors, also known as GPCRs. The hormone is a signaling molecule, or the first messenger. And the binding of a hormone to a receptor forms then the second messenger within the cell. There are two types of signal transduction pathways, both which use internal plasma membrane protein complexes called G-protein, also known as GPCR. It changes from an inactivated form to an activated form upon binding of a hormone. The features of GPCR include their integral membrane protein that span on both sides. They have many domains, E1, E2, E3, E4. On the extracellular side, you're going to have a site that binds onto the hormone, and on the intercellular side, you're going to have a region that activates and interacts with the G protein. G proteins always have three parts: alpha, beta, and gamma intercellular components. Once the G protein is activated, typically the alpha subunit breaks off and activates some effector enzyme that will turn on the second messenger. When the second messenger is created, it will then activate some enzyme that is inactive to the activated form. So the first type is called the adenocyclase. In this case, when the G protein is activated, the alpha subunit breaks off and activates an enzyme called the adenocyclase. Adenocyclase will take ATP, convert it into cyclic AMP, and with cyclic AMP is the second messenger. That in turn will activate protein kinase A and cascade a series of metabolic activities. Another G protein system is called the phospholipase C pathway. In this case, another alpha unit is going to break off and activate the phospholipase C effector enzyme. This will then split a membrane fat compound called PIP2 into two submolecules, diaglycerol, DAG, and IP3, inositol triphosphate. DAG is a second messenger, just like cyclic AMP. It will activate other kinases and therefore activate and phosphorylate other metabolic reactions inside the cell. IP3, however, will go and target the intracellular calcium storages and increase the calcium ions inside the cell. Downstream effects of GPCRs, the subunits of our G protein. G proteins have alpha, beta, gamma. Within the alpha subunit, we have three subclasses. We have the alpha S, alpha I, and alpha Q. Alpha S subunits are going to be the one that activates cyclic AMP by turning on adenocyclase and then creating the secondary messenger. Alpha I's are going to inhibit the adenocyclase and actually not produce cyclic AMP, so it inhibits the production of the secondary messenger. Alpha Q is going to be the one that activates the phospholipase C, PLC pathway, by breaking down phosphatidyl inositol bisphosphate into IP3 and DAG. What are the effects of cyclic AMP? Cyclic AMP's activity is to turn on the protein kinase A and eventually gets degraded by phosphodiesterase after the signal begins. The effects of DAG and IP3, as mentioned, is that DAG will activate another protein kinase C, whilst IP3 will target the calcium storage inside the cell and cause calcium ions to increase, changing the polarity of the membrane. In a 1-TMS receptor, for example, the receptor for insulin, it has an alpha subunit and a beta subunit. When the insulin binds, it activates a kinase activity that directly autophosphorylates different sites on the intracellular side. The P's here represent the phosphate groups that are activated on the intracellular side and activates without the help of a G protein. This will then allow the phosphate to target a substrate protein and turn that on into the activated form. And that will then trigger a cascade of signals inside the cell. Hormone receptors are nuclear receptors. Nuclear receptors are the ones found within the cell, and they serve as a transcription factor. They directly target the DNA and increase transcription. When a lipid-soluble hormone comes along, it's going to be able to penetrate the cell membrane without any problem, and it's going to target an intracellular receptor located in the cytoplasm. This intracellular receptor is typically going to be bound to an HSP, which stands for heat shock proteins, and when that lipid-soluble hormone binds to it, the heat shock proteins are going to be released. This will then cause the active monomer to dimerize with other receptors that are activated and be translocated and brought into the nucleus of the cell. This will then bind onto a section of the DNA directly, causing it to increase or decrease transcription. Therefore, you're going to create more gene products and more protein or less protein. There are some diseases that are caused by mutations in these hormone receptors. Let's take a look. Graves' disease, also known as hyperthyroidism, is when there's mutation on the thyroid-stimulating hormone receptor. Another one called Jansen's metaphyseal chondrodysplasia is a disease in which the mutation occurs on the parathyroid hormone receptor. Ovarian degenesis type 1 is when there's mutation on the follicle-stimulating hormone receptor. Familial glucocorticoid deficiency is when there is mutation on the adrenocorticotropic hormone receptor. Familial male precocious puberty is caused by luteinizing hormone receptor mutations. And then familial hypocalciuric hypercalcemia is when there's antidiuretic hormone receptor mutation. And last but not least is with hypogonadotropic hypogonadism is when you have the GnRH receptor mutation.